Okay, today we have two conferences, I think, well, two or one plus or something, two, yeah. And we have with us Henne Holstege. Henne Holstege, many of us listened to her in Lisbon in 2024. We enjoyed very much her presentation of the Centenarian cohort, the Centenarians Plus or 100 plus, how do you call
100
it?
plus study.
100 plus.
Yeah.
That is interesting, you will see. And then, well, she comes from the Amsterdam University Medical Center, yeah, the Alzheimer's department, but she's also attached to the Leuven University, where you were before, maybe, or no, that is recent. Okay, she's a genetist, and well, she will tell us about this cohort and her ongoing research. And Alex Salazar, we know him a bit better, a bit more personally, because he came some weeks ago, and he's going to stay with us 2 months or 1 month. No, 1 month. And then he will come. And we're starting to collaborate with him. Well, just anecdotically, we started here years ago, Centenarian, nonagenarian Centenarian cohort. We tried to do that, and we almost managed to do that. It was called CIEN is 100 in Spanish, CIEN. So it was, our cohort was named 90 plus CIEN.
90 plus
so it was
100.
So it was a nice name, but unfortunately our scientific director at that time was not Pascual, was a basic researcher, and he believed nothing in centenarian research. It's anonymous, I mean. So therefore we could have by now an old cohort in Madrid, and we had lots of nursing homes coordinated, but that's it. You have it, so you tell us, and we are very happy to have you both here.
Thank you very much. Can everybody hear me if I speak like this? Okay. Well, I'd like to actually comment on that because your former director is not the only one who do not believe in it. In fact, when I started this study, I call it the first 5 years, I call it my cup of poison. All the grants I submitted, every single one of them was declined because nobody believed that you could really learn something from centenarians. So he's not the only one. Well, if you could call it that. Once I showed that the centenarians are genetically protected against Alzheimer's disease, then things started to change. And yeah, but nevertheless, it's something that we no longer have to believe in, because this is all science, right? This is not a religion. And we are looking at empirical evidence. So I'm very happy to be able to show you empirical evidence to convince you that we can actually use these individuals to our advantage and really learn from them. Yeah, I think that's, so he's not the only one who was doubting this. But the world has changed, thank goodness, in our favor. So first I'm going to show you, my group. So this is my group in Amsterdam, and this is my group that's just started in Leuven. But what's happening in Amsterdam is known in Leuven, and what's known in Leuven is known in Amsterdam. What's happening in Leuven is known in Amsterdam. These are integrated groups. And the reason for this is that I'm super happy with my group in Amsterdam, but in Leuven we have more facilities to really generate novel data, specifically the omics data that is now everywhere, but you do need the newest machines, the best technologies, and that's what Leuven really invests on. It's called the VIB, so it's the Vlaams Institute of Biotechnology, and the biotechnology is really there. And I now can leverage both ends of the, or actually the best parts of each side. So I'm very happy to have this, these two joint labs. There is a open position for a senior bioinformatician. So if any of you have interest in your bioinformatician, don't hesitate to ask. Okay, so now I'm going to start with the presentation, and this is my source of inspiration. And if you've been there at the ADPD 2024, then I showed you her as well, Mrs. Hendrikje van Andel-Schipper, who was born in 1890 and died in 2005. So she was 115 years old when she died. And her mother was also 100 years old when she died, and she too, well, and one of the most important things about those really old individuals is that they are both cognitively completely healthy. And for me, this was, so she died in 2005 when I was still doing my PhD. For me, this was like a game-changing situation where it's like, wow, so 115 and no decline, that's amazing. But she shows that for us humans, this is possible. And this changed because I was actually doing my PhD in breast cancer. And I then thought, I'm going to change around. I'm going to do neurology and look into how it's possible to maintain cognitive health. So indeed, it is possible to reach extreme ages without any signs of cognitive decline. Question, of course, is how did she do it? And even though, oh, can you just go like this? Mrs. van Andel-Schipper was, of course, a wonderful person, and we did actually really learn a lot from her. But to really understand how it is possible to maintain cognitive health until a really late age, we have to compare her with other people just like her. And we chose individuals older than 100 and not demented, because after 100 years, the incidence of dementia becomes higher than the incidence of death, mortality. And that means that somewhere after 100 years, there should be nobody left who is not demented. And if you do the math, that should be about 105 years. So there should be nobody after 105 years who is not demented. That means that the incidence of dementia actually decreases, or actually, I should say, decelerates after around 100 years. And well, she clearly shows that that is, that she's clearly older than 105. So there is a change there. People become enriched with genetic factors or lifestyle factors that are protective. And we want to know what they are. So we decided to collect centenarians. We now have 520 centenarians included in the study, and we collect all sorts of biomaterials from them, including a neuropsychological test assessment, and we do this every year. So that we know who maintains cognitive health, how is the cognitive health actually, what kind of cognitive health do they actually have, and are there specific domains that decline with increasing age after 100 years old. Then we collect blood samples each time we visit them, and we do genetics, and we look at the immune system, and we look at the blood-based biomarkers, and we generate iPSCs from these individuals. Now, when we include the centenarians in the study, we ask them to consider brain donation. And about 1 in 3 of the centenarians agrees to doing so. So now we have about 140 brains that are associated with that are already donated, and there are about 34 individuals who intend to do so. And so we can look at the neuropathology and look at all the omics. Okay, so again, we now have 140 brains donated, and this is what I'm going to be talking about today. So this is the ages of the brain donors at death. This is about 140 brains, and you can see that we usually include the centenarians at 100, and the inclusion criteria is that they have to self-report to be cognitively healthy. And so we include them around here, but then of course they don't die the next day, and lots of things can happen in the years following the inclusion. So since there's at least a couple of years usually between inclusion and brain donation, I will now show you what has actually changed. Because they were healthy when we included them, but this is the mini mental state score, and is everybody aware of the mini mental state examination? Perfect. So then you know that this is a pretty high score to have at 100 something, but then there's also a group that now declined. And this for us is actually very interesting, because now we can correlate what we see in the brain with how they performed. And so we now have an interesting cohort, because the molecular changes in the brain do associate with cognition, But we also have these first steps of decline. Usually people in the brain bank are all here. So you kind of miss this change from here to here. But we now have it going from, let's say, 30 to, let's say, 20. And that is something that's pretty unique for this aged cohort. So what is our research question? Well, we all know that with increasing age, we build up amyloid plaques, and that at some time during that aging, that actually leads to the spreading and the accumulation of tau tangles. That is the amyloid hypothesis that we all work with and that we all know. Our question is, which natural protective barriers do centenarians use to maintain brain health, and what can we learn from that? So that's what I'm going to be answering today. And I'm going to show you data that's generated by these individuals. Jeroen, it's actually, because I am not a neuropathologist. I should say that up front. So I work with neuropathologists. Jeroen is the one I worked with previously, and I'm sure you know him. He transferred to Roche recently. So I now work with Vera Wiersma, and these are the PhD students that are involved with the study. And the data I'm going to show you today is generated by Suzanne Rode mostly. So, of course, we are all aware of the Thal phases for amyloid, and we all know that first the cortex is affected, and then with each phase, a deeper part of the brain region is affected. However, only 2 or 3, maybe 4 plaques in that deeper brain region will allow you to go from one phase to the next. So this is clearly a measure of amyloid spreading, not of loads. And we wanted to know whether or not the loads and the spreading were different in centenarians, because they did maintain their brain health. So this is what I'm going to show you today. Does the amyloid beta distribution regulate cognition in centenarians? That's the final question. And so we wanted to know whether or not the distribution here in the cortex, how that associates with cognitive decline. But first, we wanted to know to what extent they were actually affected and how that associated with the spreading. So now we have for each of these brain regions, we have a part, and we look at the cortex, we stain for amyloid, and we basically calculate the percentage of this yellow part that is actually affected by amyloid. So you have a percentage. And then this is what we see. We compared it with 27 Alzheimer's disease patients. We go from the frontal, well, actually, so this is the percentage here on top. And here, this is 40% positivity for amyloid. And you can see that the frontal lobe and then the parietal, temporal, and occipital parts are sequentially affected by this amyloid. It goes from here to here. Now let's look at the centenarians. And this is what we see. We see that some centenarians have a distribution that is very similar to Alzheimer's disease patients. And here we have a part that is actually, well, very limited in the level of amyloid that they have. So this part is very limited, and we can say, okay, well, these individuals were resistant to the accumulation of amyloid. But these were either resilient if they are not affected by cognitive decline, or they are affected by cognitive decline. So which is it? So now if we look a little further, we can actually also now compare the Thal phasing, and as expected, these Alzheimer's disease patients all have very high Thal phases because the spreading is all over the brain. But then here you see actually that indeed here there's also higher spreading or more spreading, but here you see a state where there can be high spreading, but very low levels or loads of amyloid. So then we actually wondered, well, so that's what I wanted to show here. So the amyloid loads can stay very low, but in some cases you already see spreading. Does the spreading occur before the load increases? That's one question. Now, how does it associate with the cognitive performance? So here we have the loads of the amyloid in the cortical region, and here we have all of the different neuropsychological tests that we administer. And then when you look here, you see that this part is a digit span backwards, clock drawing, and key search. You see that these are, or especially these two, are affected. Meaning then this is actually a correlation. So this is a negative correlation, meaning higher levels of amyloid, lower performance. And now we see that the lowest performance here is in these two tests, meaning that if you score bad on these tests, you are likely to have amyloid in your brain, or more likely to have amyloid in your brain. And this is especially executive functioning. So in this specific cohort, in these first changes of cognitive decline, executive functioning becomes affected when you have higher loads of amyloid in these cortical regions. Now, if we correct for Braak staging, then we still see that specifically this digit span backwards is actually most affected. But we also see that having Braak, including this amyloid, does actually explain at least part of this association. But what we now see is that the healthiest centenarians did resist the accumulation of amyloid. And if we now take these different tests and we say, okay, we have no amyloid, low amyloid, or high amyloid, and we take the scorings of these tests, then you clearly see that individuals with no amyloid score higher than individuals with high amyloid. And for all of these tests, the low amyloid is in the middle. So clearly there is an association here. So if we now would have to put the centenarians in a pie chart, we would see that 9% of the centenarians have no amyloid beta whatsoever, and 56% have low amyloid beta, and 35% of the centenarians are more likely to be declined because they have more amyloid and likely also a little bit more tau. So this is the loads. However, a subpart of this green part actually has a stall phase greater than 3. And of course, they died at this point with a high level of cognitive performance. However, we don't know whether or not they would have at some point, if they had lived longer, actually been part of this red part. We don't know that, but we should at least acknowledge this. Okay, so now a quick comparison with proteomics, and I do need to mind the time because you also need to present. So, which brain proteins are differentially present in centenarians? We wanted to compare the centenarians with normal people, but normal people have different ages, different sexes, and they take different medications. So it's a very difficult comparison to make. And this is the reason why we now compare all the centenarians with a age continuum. And here we have Alzheimer's disease patients in red, 61 non-demented individuals in blue, and then the centenarians. And now we can ask, okay, what molecular changes take place with age, and how do centenarians respond differently to that? But today I'm going to focus on amyloid and tau only, and we're going to look specifically here at the medial temporal gyrus. So we have mass spectrometry. We're looking only at the soluble fraction. In this specific analysis, we measured 3,400 proteins, and again, this is the gyrus temporalis medialis. Now, what we see here is that we have the map tau, and we took specifically for tau only the MTBR, the peptides that are part of the MTBR, the microtubule binding region. And we have for APP, we took only the peptides that aggregate into A beta. So those are the two types of peptides that we're now comparing. And they also explain most of the variability across the whole proteomics analysis. So amyloid and tau are basically guiding everything. But if we look now here at this very low amyloid level, we see that there are several centenarians in this part where there's really, really, really low amyloid, while the tau kind of remains very similar. But these are the centenarians that resisted the amyloid. Remember, this is the medial temporal gyrus, right? And here we go all the way to the top, where we see that some centenarians actually do have a higher load of amyloid there, but they are resistant to increasing tau. So we see that there are centenarians with higher levels, but it's only the Alzheimer's disease patients that go into this tau phase, where there's more tau in the brain at the medial temporal gyrus. Interestingly, when we look at the amount of proteins that associate with this, when we correct for all different types of cells, because the level of the abundance of different cells obviously change with the disease, but if you correct for all of the cell-specific proteins, then you see that the expression of only 5 proteins increases or changes with the level of amyloid in the brain, only 5. But when we look at the number of proteins that change in expression when tau starts to set in, then it's 50% of the whole proteome that we measured that is actually changed. So the disease is basically, it really sets in with this tau phase. It seems as though the amyloid changes prepare the brain for this, this change in tau. Of course, there is individual heterogeneity at this very end of life, and of course we see that there are people that kind of sit in the middle here. Overall, centenarians maintain a very low tau abundance, even though they can have a higher level of amyloid. So, does the amyloid spreading affect tau distributions in these centenarians, and does tau then drive the decline? So here we have the Braak staging, and we looked here in the temporal lobe. So here the CA4, CA3, CA2, CA1, and the entorhinal cortex and diffusive from gyrus. I'm sure you're all aware. So we look specific, and we did the same thing. We stained for amyloid and tau, and we looked into the abundance. We now have 112 centenarians, and we said they were amyloid negative if they have a thal phase 0, 1, or 2. And they would be amyloid positive if they have a thal phase greater than 3. And now we looked in specifically these individuals because these are obviously already down the path and these have not really started yet. So we're looking into these individuals with no amyloid beta or very low amyloid beta and positive, and then all Braak stage 3 or 4. So now we're looking now at the medial temporal or the temporal lobe in absence of amyloid. And then we see tau, specifically in the CA1 and the CA2, a little bit in the entorhinal cortex. But this is in the absence of amyloid. And actually, if we take Mrs. van Andel-Schipper's brain, because she actually did donate her brain, we see also a little bit here actually, but she had no amyloid either. And we call this primary age-related tauopathy. So this would be a representation of part. And actually, if you look at this, you can also see that in the temporal lobe, individuals with Alzheimer's disease have higher levels of amyloid. And so this is the white here. And these are all these regions going from here to here. And you see that the centenarians have lower levels here. But this is actually, I think, 10-fold lower than what we see in the cortex. So it only really starts to reach this at the very end. And then this is how the amyloid spreads. So you see the fusiform gyrus and then the entorhinal cortex, and then it starts to sit here. So now we can look into with tau in the presence of amyloid, and that's what you see. So when the amyloid kind of comes in here, it kind of changes the positioning also of the tau, because this is thal phase greater than 3, and now the tau is also here. So if we move this, what was just here up, and we now also project the amyloid positioning here, then we see that where the amyloid is now also, compared to that one, where the tau has transferred to, as if it kind of just moves in and then pulls out the tau from here into here. Because you see also that it's that there is no amyloid in this part. So we think now that we wanted to ask now whether or not the amyloid presence or the tau presence associates with cognition. And then first we did this. So we have now the amyloid loads in these different regions and with these different cognitive performances, or these are the cognitive domains. And we have the tau here. So higher levels, redder, lower performance. And we see that with amyloid and with tau. Now, if we look here at the top, this is really the hippocampal region. You see that actually higher levels of tau, even that doesn't matter at all. These levels don't do anything with cognition. So this part does not associate with cognition. But if we look at the lower part, the perihippocampal region, we see actually that there is an association with cognitive performance here. Perihippocampal amyloid and tau does associate with cognitive decline. But again, we can now correct this. We can correct amyloid levels for tau, and we can correct tau levels for amyloid. And if we do that, so we see really only the effect of amyloid and only the effect of tau on cognitive performance, then this is what we see. It actually is almost gone here. So whereas here it's actually stronger. And this means that, yes, there has to be amyloid in this part, but the actual cognitive performance is regulated by having tau there. So parahippocampal amyloid and tau do associate with cognitive decline, but it's actually driven by the level of tau. So now what? So we have amyloid negative and amyloid tau positive individuals, and their cognitive performance, and we take here global cognition, is very similar. But if we take this group, this amyloid positive group, and we divide it into tau positive and tau negative individuals, then we see that it's the amyloid positive and tau negative that actually has a very similar cognitive performance. But the individuals who are amyloid positive and tau positive, they are starting to decline. So now we have this interesting group that is amyloid positive and tau negative. That's actually pretty interesting because it explains a large part of the centenarians. So there's a unique group of amyloid positive centenarians that keep the tau abundance specifically low, and they maintain cognitive performance. Now, if we look at the amyloid levels in these individuals, sometimes it's a pretty big level of amyloid, and the tau is really comparatively very low, because there's always a comparison between the level of amyloid and tau that kind of belongs together. But in the centenarians, it's especially low, the tau. So now this moves us into this one. So we have about 40% of the centenarians resisted amyloid, and so basically they maintain a very, very young brain. There's 30% of the centenarians that were resilient to amyloid, but resistant to tau. And there were 30% of the centenarians that had higher levels of amyloid and tau, and those were the ones to be more likely declined. And I think this is interesting because this resilient group is interesting. Amyloid, no tau. And that's very similar to what we see in the Christchurch mutation. They also had amyloid, but very little tau. Colbos Wheeling mutation, also a lot of amyloid, no tau. So these are individuals with a presenilin mutation, right? So they have a lot of amyloid, but specifically low tau. So would this group of centenarians be representative of like a whole set of Christchurch mutations and a whole set of Wheeling mutation? This is something that we will now be using in our analysis. So we will be separating our whole centenarian group according to this pie chart and really looking into specific mechanisms that allow amyloid but resist tau, and individuals that might have a different mechanism where they maintain a very young brain. So that brings me to my question to you, actually. Should we now extend the amyloid hypothesis by this new state of resilience? Because it's amyloid and then tau, but it's actually amyloid, but not necessarily tau in this subgroup of individuals. And that might have actually specific mechanisms that these centenarians or the Christchurch or Colbos carriers actually use to keep their brain healthy. So this brings me to my last slide. So 70% of the centenarians were protected against cognitive decline. Of those 70%, 60% resisted the accumulation of amyloid, 40% were resilient against it, and 30% have actually more amyloid and more tau after we included them, and they were more sensitive to develop decline. We see that specific in the amyloid in the cortex associates with executive function, change in executive functioning. Part is very prevalent in centenarians, but does not associate with cognitive decline. Amyloid beta kind of comes in from the temporal lobe to the fusiform gyrus and the entorhinal cortex, and then also p-tau comes in. And then Alex is going to show how this may be genetically defined, because usually I would also present some of the genetics, but Alex, the person who's actually doing this, will do that here. And so now we have next steps. We will link the genetics to these differential mechanisms according to how the centenarians actually looked phenotypically in terms of amyloid and tau. So we're now doing single nuke RNA-seq, proteomics, and lipidomics, and to compare that with neuropathology. And now, yeah, to finalize, and I'd love to talk about that when we close the seminar, but should we now extend the amyloid hypothesis with this new state of resilience? So with that, I'd like to end this part. And this is what the centenarians say themselves. And I'm sure you saw this slide at ADPD, but it's always fun to show it because this is the people that we're learning from. And of course, all the people that work with this study, and these are the centenarians here that we celebrate every year, because we do get to visit them every year. We do get to draw blood from them. We do get to do all these comparisons. And if we find something interesting, we can go back and get more blood. So for us, this is a collaboration between us as researchers, and they are the persons who are being researched. But the fact that they are so welcoming to us is well, heartwarming, and that we celebrate that every year. So with that, I'd like to end and give the presentation floor to Alex, who's going to explain how this is all genetically defined.
Thank you.
Alex, are you?
So I think it's we let Alex explain present his talk, and then we have our questions. Yeah, just not to stop.
Yeah.
Okay.
Oh, okay. So then I will stop sharing, and then you can log in, and I will have to give you the microphone. Okay. Logging. Logging.
Move it over here. Yeah. Great. So I'll pick it up from here and Sabin? Okay. Yeah, well, I'll pick it up from here and talk about genetics. As Hannah was alluding to, centenarians are enriched with genetic protective factors. But to maybe paraphrase George Orwell a bit, although all the AD variants are equal, some are more equal than others, and it turns out centenarians are very specifically enriched with some parts of the genome, and that's what we think makes them special, and perhaps that's actually one of the mechanisms that are driving these signals that Hannah was talking about. So we're going to start very broad first. We're going to look at just how, in general, centenarians are enriched, and then we're going to zoom into the specific parts where they're particularly special. And I broke this down into 4 parts. I'll first start about just give some backgrounds on the previous findings that we see. It's just going to be a quick summary just to set up the stage. I'm not going to go into the major details, and then we'll dive into the genetics. We're going to zoom into one very specific gene that is very particularly strong in centenarians. We'll see how that actually links to pathology, particularly tau and TDP-43. And if there's time, we'll also talk about mechanisms, because as Hannah's saying, we want to understand exactly how you go from genetics all the way down to pathology. We have started to do a little bit of work. This is my idea. Let's see if I can fit it in, because I put all these slides together and then I wanted to start to trim things down. But of course, there are other priorities. So this is my daughter at the Parque de Retiro. Well, as you can see, she thinks there are more important things to do than to work on these things. But, let's see how far we can get this and how quickly we can get through this. I don't think I need to explain to you about neuropathologies, but to me, I think one of the things that has always been very interesting is that even though we often see these neuropathologies as hallmarks to these different neurodegenerative diseases, of course, they don't exist in isolation, right? And then a good example of that is in Alzheimer's disease, where the primary pathologies, although they're amyloid and tau, we definitely see people who go on to develop different co-pathologies, including TDP-43 and alpha-synuclein. And so the work that I've been doing and what we've been doing in the group as well, overall is in something that's still very much an open question, is that, well, what exactly, besides age, are the risk factors that pushes people towards this pathology trajectory. Hannah just talked about the Centenarian study, and just to also quickly summarize the work from our PhD student, Susanne Rode, Hannah just mentioned about the resistant and resilient centenarians, right? Centenarians that develop little to no amyloid tau, but also centenarians that do develop these pathologies, but seem to somehow escape those negative effects. Now, the paper and also what Hannah presented was mainly about amyloid and tau, but we do have to say that these groups also extend to other co-pathologies, because we also see centenarians that develop little to no TDP-43 and alpha-synuclein, and we also see centenarians that do develop them, but again, stay cognitively healthy up until death. So of course, one big question that we have at the moment is, well, how is this possible, right? What exactly are the mechanisms that is allowing these individuals to escape all of these age-related neuropathologies? So as I mentioned, we think that at least part of this is being driven by genetics. And what you're seeing here on the slide is the classic Manhattan plot, which shows you the genome-wide associations of Alzheimer's disease, showing you in this particular plot 91 different regions in the genome that are associated with Alzheimer's disease. And a bit of a funny coincidence, but also bad timing. I think last week there was a talk about polygenic risk scores, but I couldn't actually make it just for bad timing because the person was actually from Amsterdam. I think I also saw Emma that's online. But one thing that we can do, and what we have actually shown before, is that you can build these polygenic risk scores. And if it's still a little bit confusing, all you need to know is that it's just an average, it's a weighted average summary, average score of risk. So you take all these 91 loci, you look at what is the strength of risk for becoming a risk for Alzheimer's disease, and then per individual you just simply calculate what is the sum of the score. You look at the distributions, and in this case we calculate this PRS in the centenarians, compared them to a cohort of 80 cases compared to a cohort of controls, and overall what we basically see is that centenarians in general have about a 2 to 4-fold lower risk for Alzheimer's disease in aggregation of the entire genetic region, right, the entire genome. Now, of course, the thing about these type of analysis is that, well, we're particularly interested about mechanisms, right? So are there is it that every single one of these variants is really doing something equally, or are there some of these variants or some pathways that are much more strongly common in the centenarians? And again, one way to see this is just by asking the simple question, right? Are all variants equally enriched in the centenarians, or are there some of these protective factors that are more enriched than others? So to quickly summarize these results and not to go into the details, otherwise we run out of time, if we do this enrichment analysis, we looked at the top two hits, the first and the strongest hit shouldn't be too surprising, and that's ApoE2, right? We know that this is by far the strongest protective factor that in Alzheimer's disease when it comes to genetics. But the next strongest signal that we see is actually in a gene called TMEM106B. Now, some of you may know TMEM106B, particularly if you're from the neuropathology side, and that is because TMEM106B itself is also pathology. It's actually a fairly recent pathology, only a few years ago was described to, on top here you see the entire protein, the TMEM protein. It's not that big, it's only 274 amino acids. And the part in yellow that you see here highlighted, well, that part actually aggregates. It forms fibrils and aggregates in the human brain. And it has actually been seen in all sorts of different brains, from just regular age brains, individuals with Alzheimer's, Parkinson's, FTD. It seems to be in basically neurodegeneration in general. And for the last couple of years, it's actually been an open question. Are these fibrils actually just reflections of a neurodegenerative process, or are they benign? Or are they themselves also drivers of neurodegeneration? And just over the last few weeks, there's these two recent preprints from Michael Wirtz and Leonard Petruccelli's group, actually showing that, well, it turns out that these fibrils are actually neurotoxic, right? So perhaps one of the things that at least we're starting to do now is to also look at exactly this TMEM106B fibril pathologies in our brains. But it does show that, well, there are more pathologies to look at, and we should definitely start to consider these things. Now, for us, at least, when it comes to the genetics, what's particularly interesting is that if you zoomed in to the TMEM106B locus, right? And so here is basically the GWAS. If you've never seen this, it's just the GWAS summary. On the bottom, you see the T106B gene, and every dot is a variant. So one dot here is a variant, and on the y-axis is just how strongly associated the single variant is with Alzheimer's disease. Now, the variant that the centenarians are enriched with is here in the middle. That is again, the second strongest signal that we see in centenarians next to ApoE2. But you'll see that this variant is not necessarily by itself. It's actually part of this other block in red. That's what we call the linkages equilibrium block, because that actually represents a piece of sequence that we all commonly inherit. So if you have the variant in purple, you also have the variant, all these other variants in red, right? So it's not that you inherit one individual variant, you actually inherited this whole set of sequences. And the interesting thing about this piece of sequence that is commonly inherited in the population is that it's already been associated with all sorts of different neurodegenerative traits, whether it's risk for frontotemporal dementia or risk for TDP-43 related pathologies like hippocampus sclerosis and late, risk for the abundance of the fibrils that I just talked about, enrichment in centenarians, risk for Alzheimer's disease, as well as risk for the spread and burden of tau. And so one interpretation of this is, well, maybe, perhaps, one of the reasons why there's such a strong signal of this locus in centenarian is that this is a locus that is somehow involved in all of these neurodegenerative processes. Now, we do have to just make a quick note here that one very specific variant on this piece of sequence that people normally inherit is actually a coding variant. So threonine to serine amino acid, so it does change the protein. And there's been a lot of debate about whether this single variant is actually the sole driver of all of these effects that you see. It's still very much debated about what's the thing, about whether this is indeed, even though we have a lot of variants where all of these effects are functional effects, at least, are being driven by this specific substitution. But what we see is that even though there's this specific LD block, when it comes to Alzheimer's disease, there's actually a second genome-wide significant LD block. This means that what you see here on the bottom, that is a completely different sequence that we are commonly inheriting that contains a completely different set of variants that is different from the ones that you see on top. Now, what exactly does this mean? Well, I'm going to take a quick detour and actually talk about genetics of ApoE, which is, I think we should all more or less be familiar, at least with ApoE, right? We know about ApoE alleles E2, E3, E4. Generally, we say that E3 is the neutral allele, so that's the baseline allele, and then E4 is the risk-increasing allele, and E2 is the protective allele. Now, ApoE is not a single SNP, right? ApoE is actually a haplotype. Or in other words, it is it's a commonly inherited sequence. And on that haplotype is actually fairly big. There's all sorts of different variants, but the two variants that are particularly important in E2, E3, and E4 are actually just two SNPs that are coding variants. And these two SNPs change the amino acid profile. So you actually, depending on whether you have E2, E3, E4, well, that means you have a different combination of amino acids. Again, these are not the only SNPs on that haplotype, but these are the ones that people pay attention to most. And in theory, technically speaking, there is also E1, which is a very specific combination of these SNPs, specific combination of these amino acids, which is theoretically possible, but we just don't see it in the human population, probably because it's very deleterious or has been selected out of the population. Now, the reason I talk about ApoE, which again, we're talking ApoE in the end, E2, E3, E4 is a haplotype, right? It's a combination of variants. The reason I mentioned this is because when it comes to T106B, the fact that we have two completely different LD blocks, this is actually suggesting that something similar is happening here. That at the end of the day, the genetics of this locus is actually about the combination of all these different variants, not necessarily the inheritance of one. And so what we've done in our data, in our cohorts, is we've done something called long-read sequencing, right? So when we do these type of GWAS analysis, the way GWAS works is that it's an array, you genotype different variants, and then you predict the presence of other variants. But those types of technologies only allow you to predict individual SNPs or small variants. And the nice thing about long-read sequencing is that you can also identify small variants that you typically see in GWAS, but you also pick up signals from variants that you don't normally see. Those are particularly structural variants, large genomic rearrangements, large structural changes that you do not see in GWAS, as well as DNA methylation. So you also see the epigenetic levels of the DNA sequences that you're inheriting. And particularly, the more important part here is that we can actually do the haplotype reconstruction, which is called phasing. In other words, we can reconstruct the sequences that are actually being inherited in these genomes and actually see, well, what exactly are the haplotypes in this TMEM106B locus? So what we found is that when we do long-read sequencing, we indeed see that there are not SNPs, right? But there's actually haplotypes. And specifically, there are 4 distinct haplotypes in this locus. So up here, I have a small cartoon. You see the TMEM106B gene. And this locus actually extends out all the way up front, where we particularly see this cluster of distal enhancers. So these are regulatory elements upstream of the gene. And just to walk you through these haplotypes, we call them T1 through T4 in descending frequency, the most common one being T1 at 57%. And the least common one, which it's fairly rare, but it still exists, at least in the population, is something called T4. That's about 1%. What's specific about these haplotypes? Well, what we see is that the most frequent version actually contains this threonine amino acid, along with a retrotransposon in the 3' UTR, as well as a very specific methylation signal that is happening in the intronic sequences. So that's what we see with T1. When we go to the next haplotype, we see a haplotype that generally only includes the serine amino acid, right? So it's just the other amino acid substitution. We then see another haplotype that's similar to T2, which also has a serine amino acid, but instead has a large, a specific set of structural variants, of actually big genomic rearrangements, specifically occurring in this site of distal enhancers. And then the last haplotype that we call T4, that's actually a recombination between T1 and T3. Now, I just told you that the second strongest signal in the Centenarians is this TMEM106B gene, but that was based under the assumption that all that signal is contained within a single SNP. But you can clearly see that that's not, right? It's actually haplotypes, it's combination of alleles. So, of course, one question that we had is, well, in the context of these haplotypes, where is the signal coming from, right? Is there a specific haplotypes that the Centenarians are specifically enriched with? So what we did is we took our Centenarians, we compared them to a cohort of non-dementia controls, over 3,000 of them. And then what we see is that there is indeed differential enrichments of these haplotypes in the Centenarians. What we specifically see on this plot here, what you see on the left, on the y-axis, is just the different haplotypes. And on the x-axis, that's just the odds ratio of being a Centenarian relative to middle-aged non-dementia controls. And what you can see in this comparison is that Centenarians are highly depleted with the T1 haplotype, which is the one that you see in red. They're intermediate for the T2 haplotype, which is the one you see in blue. They're highly enriched with the T3 haplotype, which is the one you see in purple. And T4, since it's relatively rare, you can see the wide confidence intervals. It's a bit hard to say what exactly is happening there. And again, I mentioned before that for the past nearly 15 years, there's been a lot of debate whether at the end of the day, all of these effects are coming from the threonine or the serine, right? That even though you have all these variants, it's really just these two amino acid substitutions that are driving the signal. Well, one thing we can do is we can do a baseline, just like in ApoE. We set the baseline, the reference to ApoE3. We say that that's the neutral. We can do something similar here. We say, okay, so we baseline to T2, which only has a serine amino acid. Do we still see enrichment? And indeed, that's what we see. We still see that relative to the T2 haplotype, there's still a 1.3-fold higher odds of being a Centenarian if you carry T3, and much lower odds if you carry T1, basically saying that all of these other variants, right, these like particularly the structure variants, those are making a difference, at least in the Centenarians. So this is the first part, right, and then that's the genetics. And I think, and as I just showed you, there are at least 4 distinct different haplotypes in TMEM106B, one of them being very strongly enriched in Centenarians. And of course, the next question is, well, what is it actually doing, right? So there's clearly a preference of these haplotypes in the Centenarians. How does this actually translate to pathology? So to do that, we wanted to basically look at the haplotypes and then correlate and associate those haplotypes with pathology. As I mentioned before, this specific locus has already been associated with tau and TDP43 as well as TMEM106 fibrils. We don't have data for the TMEM106 fibrils yet, so we'll just look at tau and TDP43. And I'll first start with tau pathology. So what we did is we have this cohort. We call this the 100 plus MBB cohort. It's a cohort of nearly 200 brains. It comes from brain donations from our very own centenarians in the 100 plus study, as well as brain donations from the Netherlands Brain Bank, including 80 cases and controls. And what we did is we took this data, and what we did is we asked, well, do these haplotypes have an effect on tau spread, particularly using Brock staging? Right, so do these haplotypes actually make a difference when we look at tau spread? And what we did here is, again, we baseline to the T2 haplotype. So this is only the haplotype that carries the serine amino acid. If you run this association in this cohort, what we see, and that is on the y-axis are the 4 haplotypes, or the 3 haplotypes, because this is baseline to T2. On the x-axis, you have the odds ratio for higher Brock stage. You actually see that the haplotype that the centenarians are highly enriched with, that is actually significantly associated with lower spread of tau. And this is an effect that still remains, even if you remove the centenarians, only compare 80 cases in control, indicating that this is not an effect that's being driven only by the centenarians. Now, of course, this is only based on Brock staging. One question is, would we still see the same effect if we use another pathological assay for tau? Well, Henne just talked about the fact that for the same cohort, we did proteomics, right, specifically in the middle temporal gyrus. So what we did is we took the overall abundance of the microtubule binding region, which is, again, it's the region of the tau protein that is prone to aggregate. We took the region of amyloid beta, so the amyloid beta peptides, and basically what we ask is, do these haplotypes also associate with the abundance of the microtubule binding region adjusted for the abundance of amyloid? And again, if we run the same associations, we still see that when we looked in the middle temporal gyrus, when we look at the abundance of these proteins, we also see that the same haplotype that the centenarians are enriched with is also associated with lower abundance of MTBR tau. And again, it's an effect that we see even if we remove the centenarians and only compare 80 cases and controls. Okay, so we see that there's at least one haplotype that has an effect on tau, but what about TDP-43? So one of the issues with this cohort, and I think as Alberto, you mentioned before, that sometimes new pathologies come, and then you have to make an effort to go back and redo the pathologies, right? So here we actually, for very similar reasons, we don't have TDP-43 information for all the brains, right? They're maybe, I think for maybe 2017 or 2018, 19, they just started to do TDP-43, so actually for a lot of the older brains, we don't have that information. So instead what we did is we went to another cohort, and that is the ROSMAP cohort, or the Religious Order Study and Memory Aging Project, that has a lot more brains, over 1,000 of them, have a few centenarians, but mainly 80 cases and controls, and basically we did a data request to get those brains and also get the available TDP-43 staging, which is available for all these brains. And then basically the exercise here is, well, using this different cohort, what are the effects of these haplotypes, except now on TDP-43 spread? Well, if we run exactly the same association models, but this time looking at TDP-43 spread, we actually see that the haplotype that the centenarians are depleted with, right, which is a T1 haplotype, well, that's actually associated with higher spread for TDP-43. And this is, again, an effect that we also see even if you remove the centenarians from this specific cohort. So what does this actually mean? So we take a brief minute just to summarize, talk to you about these 4 different haplotypes. And that depending on which haplotype you're looking at, one of them, T3, we saw is highly protective against tau. The other one, T1, is risk increasing for TDP-43. And similarly, when we do this ApoE type of analysis, we always baseline to ApoE3. In this case, we baseline to to T2. But of course, you can change the reference. You can always have this relative to anything else. And I won't go into the details, but one of the things that we did is we just looked at the overall effect of these pathologies, regardless of what you carry. And one of the things that we actually see is that in general, carrying T2 is actually risk increasing for tau, and carrying but at the same time, risk decreasing for TDP-43. Now, if you look at this association metrics, right, where red is risk, blue is protective, and you look at it for a second, you start to realize there's a bit of a pattern here, and that is a bit of a tau TDP43 dilemma, right? Where according to genetics in this single locus, it's either pushing you towards having less risk for tau at the expense of TDP43, or inversely, less risk for TDP43 at the expense of tau. And funny enough, this is actually a recent discussion. This is published work from Dorothy in Germany, showing in vitro that tau fibrils and TDP43 fibrils actually physically interact, such that, at least what she sees, is that the TDP43 fibrils lower the seeding of tau at the expense of TDP43. And of course, what we show here is that, well, there seems to be actually a source to this, and there seems to be a genetic source to this specific interaction. And if we now think about the centenarians, right? Well, we saw there's a very specific pattern that the centenarians are highly depleted with T1, highly depleted for T3. Well, we put it in this context, this is actually the optimal combination for lowering risk for both tau and TDP43, right? So accordingly here, the genetics here can actually protect you against both, which we think might actually be one of the reasons how they can reach old age by avoiding both tau-driven dementia, but also TDP43-driven dementia. So that's the pathology. But of course, one big question that we have is, mechanistically, how is this happening? Now, this part of the talk gets a little bit confusing and a little bit complex. So I'm just only going to focus on T3 because we do look at this at the other haplotypes, but for now, I'm only going to talk about or mainly focus on the mechanisms behind T3. That is the haplotype, again, that is enriched in centenarians and protective against tau. So again, we have these haplotypes. We saw effects on pathology, particularly tau and TDP43. The big question is, what is the black box, right? How is this happening? And what we decided to do here is we decided to do a multi-omic integration. We said, okay, we have genetics. So how does genetic actually rewire the chromatin accessibility in cell types? How does this affect single cell type expression? How does this affect a proteomics? And does these effects ultimately lead to tau and TDP43? And whether these effects do explain the effects that we see in tau and TDP43. I'm just going to summarize this just to make sure that I'm running time. But the first thing that we did is we looked at the chromatin profiles, because even though we looked at genomes as a linear, it's not linear, right? Genomes actually fold onto each other, and then they can actually regulate in very particular ways. And when we actually integrated chromatin profiles, what we actually see is that TMIN106B has three neighbors. It's a gene called TH70A, VVDE, and C7orf78. And basically what we see is that the DNA actually folds onto each other. And so TMIN106B is part of this 4-gene promoter hub, which means that there seems to be coordinated control, meaning that the expression, the gene expression, and perhaps even the protein abundance of one gene will be highly correlated with these other genes, which also means that if you have genetic variants that influence the expression or influence the abundance of protein of one gene, that should actually also influence the expression and protein abundance of the other genes. So what we did is we looked at single cell data to actually test that hypothesis. We actually integrated a single cell data from ROSMab and basically asked, do these haplotypes influence not just gene expression, cell type specific gene expression of TMIN106B, but also all these other neighbors in the promoter hub, and if so, in which directions? And basically, just to summarize, what we see is that we see a very particular strong signal in neurons, right? In both inhibitory and in excitatory neurons. What's interesting here is that when we look at expression of TMIN106B, the two haplotypes, T1, which is the one that centenarians are significantly depleted with, that significantly lowers expression in neurons. T2, which is this haplotype they're intermediate with, increases expression, but T3 actually maintains it. So the haplotype that they're significantly enriched with, protect against tau, actually seems to maintain expression of T106B. But interesting, when we look at the other neighbors, particularly the VVDE and TH70A, we see the biggest hit on T3. So T3 actually seems to be driving expression on these two other genes. And then when we go to the proteomic data, particularly in our proteomic data, we actually see this translated back as well. We see that specifically the T3 haplotype increases the overall protein abundance of this gene in the neighborhood, But also interestingly, the T3 seems to lower the abundance of the C-terminal part. So we don't know whether that's actually the fibrils, the C-terminal fibrils. We only know that that's the overall abundance. It could be functional TMM, it could be the aggregated fibrils, but we do see that this haplotype, even though it maintains expression, it does lower the amount of the C-terminal part of the TMM106B protein. So one hypothesis here is that there seems to definitely be coordinated control in this hub. The genetics influence the expression and the protein abundance of this hub. So could perhaps all these effects, are these effects just being mediated by how you control this 4-gene promoter hub? And so we did a mediation analysis in our proteomic data set and basically asked, are the effects on tau explained by how much TMM106B or THC70A do you actually see? And surprisingly, just to be very brief, we don't. Even if you adjust for all of these, the haplotype remains significantly protected despite of all these differences. So there seems to be something else not related to this. Even though it has a significant expression and control of this, it doesn't fully explain the protection against tau. And interestingly enough, this also seems to have not a big effect on the neuroendolysosomal system. So what we wanted to see is that, okay, if it's not the hub, if it's not the neuron endolysosomal system, well, we know that tau also spreads through microglia. So could T3 actually perhaps be somehow regulating microglia? So what we did is we looked at microglia subpopulations. So this is from the ROS-map data, and what you're seeing in this UMAP are 15 different subpopulations of different states of microglia, some of them being proliferative, some of them being stress-related, some of them being homeostatic or surveilling, some of them being inflammatory. And basically what we ask here is, in this data set, is does carrying a specific haplotype, is that associated with higher chances of being in a specific state, right? In other words, does carrying a haplotype lead to a specific cell type activation or not? And what we see is that if we run a multinomial regression model, we do see at the end of the day that there is one specific haplotype that T3 is associated. Again, this is a haplotype that Centenarians are enriched with, the one they're protective with. And it's a state called microglia 15 population is actually associated with inflammatory state. So it seems to be that if you carry this haplotype, you're less likely to have an inflammatory activated microglia. So on that note, we think that at least T3 seems to be mediating its effects on tau, although this is still very much a question that we're still trying to resolve. I didn't talk about this, but T1 and T2, on the other hand, do seem to go through this endolysosomal system in neurons. And I just want to take a brief second and actually zoom out, right? Because I just talked to you about all this complexity in this one single locus, right? So that's TMM106B. But there are other genes that centenarians are also enriched with. And one of the other signals that we see is actually in HLA, which maybe some of you may know is associated with our immune system. We've actually been doing some genetic fine mapping analysis of that. And if you want to know more about that, talk to Danny, who just on his way to finish his PhD and had his whole PhD on Pacific HLA. Because although TMM106B is a very strong signal, as I mentioned, there are other signals that make the Centenarian special, one of them being HLA. So with that, I just want to acknowledge your group, but also the Netherlands Brain Bank and the Rush University for allowing us to use their data sets. And I think we can stop here.
Thank you very much, both. I think I have hundreds of questions actually, but I will select some of them. Just a couple. No, I think you can keep your microphone and we can share this one. Well, you and the other. So just two very macro questions. 100 is a sort of a line. It's a sort of cultural line. 100. I want to be centenarian. it seems to me it's something like 65 years old for the distinction between early onset and late onset Alzheimer's. And there's some discussion about it, surprisingly, because I believe that early onset and late onset are different forms of Alzheimer's disease. But some people really believe that 65 is the most frequent until now age of retirement. So maybe someone selected 65 and then you see what before and after. So I was just thinking 100. And some years ago, I think it was more frequent to talk about oldest old. That meant 85 plus, 90 plus, including centenarians. Now there are some very interesting studies also of the Japanese with super centenarians, I guess over 110. So seems there is a way to look at it with cuts, with segments, and there is a way, I guess, to look at it as a continuous change. So maybe that your data could be interpreted as a continuous change in risk. But I think that is very difficult. I don't know if you have thought about that or.
I can definitely answer that question. Maybe I should just stand up so that everybody. So clearly this is, of course, not a black and white thing, right? This is clearly a continuum where you see that individuals with Alzheimer's disease before 65 are more enriched with genetic aspects. I mean, presenilin 1, of course, being the very much the most, but of course, also that is, there's also a continuum there. You have SORA1 mutations that if you have a protein truncating mutation, for example, that gives you relatively early Alzheimer's disease, especially if you combine it with ApoE4, for example. And so all of those stronger mutations will be more enriched in earlier onset. But that, of course, changes a little bit, with increasing age. So individuals that have Alzheimer's disease at 75 will maybe be more likely to be heterozygotes for ApoE4 and have a less strong SORA1 mutation and all those combinations. And all the way until the very end, where we see clear depletion of all sorts of genetic risk factors for Alzheimer's disease, which we see in the centenarians. Our point being, though, that is not an equal depletion. What Alex just also referred to is that not all variants were created equal. That's how you nicely introduced it, saying that some Alzheimer's disease risk factors are very much depleted, like ApoE, like ApoE4, or TMEM, but there are several others where we see clear depletion, whereas others are slightly depleted. And so there is, of course, it's a continuum. And, let's say that the age at death is usually distributed like this, right? And we are sampling here with regard to having really, really high ages at death. And if you combine that with no cognitive decline, then we're actually sampling even further. We also see that individuals who have a high cognitive performance are also the one to die later. So there's also a combination there. But yeah, we should be always viewing this in this distribution. And whereas early onset Alzheimer's disease will probably be on this side, we're sampling on this side, but within it, I fully agree, is a normal distribution, if you can call it that.
It's a gradient.
Yeah,
yeah. And I think maybe it's also worth saying that this is also why one of the things that we want to do next is the big project, which is basically we are taking 1,000 to how many brains now? But it is postmortem brains of a gradient, of an age continuum gradient, but also not just in Alzheimer's disease, but also in other related forms of dementia, because at the end of the day, you have age as a dimension, but you also have all these like an Alzheimer dimension or FTD dimension or Parkinson's dimension, right? And at the end of the day, try to disentangle what is age-related, what is Alzheimer's, what is FTD, right? I think that is particularly also feeding to it, which is going to be part of a trajectory, right? What trajectory do you end up? Do you end up healthy aging or non-healthy aging? And if it's non-healthy aging, is it Alzheimer's? Is it Parkinson's? Is it late or something in that matter?
And for that, I think it's interesting to add when we're discussing continuums. Alzheimer's disease is obviously much more prevalent than Parkinson's disease or any other types of neurodegenerative diseases. So to reach 100 years old without cognitive decline, you have to be protected against Alzheimer's disease, because otherwise you would get it, right? Because it's so prevalent. So we do see an enormous protection against Alzheimer's disease with regard to genetics, but we also see it with regard to Parkinson's disease, but much less, because you don't need to overcome that, because the chance of getting it is less. So we really are looking in the Alzheimer's disease continuum. But of course, the question, of course, lies. So remember the aging continuum I showed with the proteomics. So that's exactly what we're building on. Now we're going to 1,000 individuals, and we're and at different brain regions, because that also matters, of course. And now we can ask, okay, you have normal aging, or let's say healthy aging, but what makes you move over from this healthy aging trajectory to a PD trajectory, or an AD trajectory, or an FTD trajectory? And what genetics are necessary for that? And that is what, Alex has been extremely helpful in finding this out. In fact, I think I have to commend Alex here, because he did a lot of research that was just totally new. Nobody really knew how to look at this, and to deconvolute a locus like this was completely new, and he was really pioneering in that. And I think that's
It took
really
some time to figure this out.
it took a lot of discussions, but they were all fun to do, to have discussions. But I think you've really done a great job in figuring out this new aspect of neurodegeneration, namely how is it genetically directed.
Okay, thank you very much. Just one short question, because before I leave the others, there is a small city in the middle north of Spain that's called Soria. Soria is it's a very nice place. So Soria, yeah, Soria. So we still have a donation program in this, because there are lots of centenarians. So when we were interested in Centenarians, we started with our Soria program, because centenarians are famous in Soria. They are protected by the authorities, and every time that someone reaches 100 years, he or she get a certificate, and there is a party in the city and in the town hall, and they are received as honorable people in the city, independently of their cognitive status. So just my question, with the epidemiological genetic information that we have now, how much extrapolable are your data to other genetic populations, or genetically?
You mean to younger individuals also?
To other
or to other people? Yeah, that's a good question. This is also very much the reason that we've just embarked on a new study with Nicolo Tesi, and he is now we've approached all these different genetics, centenarian studies across the world, even Japanese and in South America, and we're going now to do the exact same study that we did with our centenarians, and to ask, are the centenarians in their study similarly enriched with the same genetic loci? In other words, is the mechanism the same or not? I can't answer that question now. Only that study can answer that question. Yeah.
And maybe on that side, it is also worth noting that specifically we do think that this whole concept of haplotypes is not just in TMM, it's actually probably in many places of the genome. And the interesting thing is actually there are some regions that are already very well known to be different in different ancestry. Actually, TMM is one of them. We actually, in our preprint, we show that as soon as you leave European population, or let's say more Central European population, you see completely different haplotypes, completely different combinations of alleles. What do they do, given that we see such a big effect? Right, and I know that the Spanish population is maybe a bit more heterogeneous than the Dutch population. So if you were to look at, let's say, that specific population, do you actually see even new signals, right? That is also possible.
And a question then to you: is the Soria population still there? Is it still enriched in centenarians? Well, why not approach them again then?
The thing is that we have some brain donations, but not of centenarians. Thank you. Yeah. Well, the thing is that, they depend on another brain bank, and so we just missed a bit, but the intention was to get brains of centenarians, and some of them cognitively, at least, well studied, and but then that was we haven't got that population, but the people are there. So I mean, we could try to reach them. Yeah,
that would be
that
nice.
would be interesting. Thank you. Yeah, Theo?
I don't have hundreds of questions. I only have many questions, but I only will ask you three simple questions. One of them has been, I think, partially addressed just now. The subgroups that you have described, the three subgroups in your population, in your cohort, could be found out also in other similar cohorts. That's something.
That's a question, right?
Yes.
I think, of course, we select. Oh, I need to get it. Yeah. So we did, of course, select for individuals who are really protected against decline. I mean, if you don't have amyloid at 100, that's kind of odd, isn't it? If you're not, and also clearly, by far, most centenarians, also in the Dutch population, are very much demented. If not Alzheimer's disease, they're very much declined. So to have this subgroup that is still healthy means that we really enriched for these individuals. Now, this specific mechanisms where you have a lot of amyloid but no tau, that of course exists throughout the whole population. But of course, we don't really see it very often because individuals that are protected by this don't go to the hospital, right? So because they're healthy. Now we have to really select it out by the Cole-Bos mutation or the Christchurch mutations. And then that's when you see them, when they are also at risk for something. Now we are looking at aging as a risk factor. They are looking at presenilin 1 carriers as a risk factor. But this is a mechanism that exists in the population. How often it occurs in, let's say, in a cross-sectional 80-year-olds, I don't know. I don't think it's too prevalent just by feeling. Otherwise, we probably would have already been more aware of it. But it exists, but we can learn a lot from it if we can actually understand what the mechanisms is that you can withstand the effect of amyloid and maintain tau low. That's, of course, very informative. That's why everybody's so happy about the Christchurch or the Cole-Bos mutation. Yeah.
My second question is about you have presented some genetic characteristics of these centenarians with healthy state, cognitive state. But do you have any other markers of these centenarians in good state? Because in a paper that we published some time ago about non-Nigerians, we found out that the GFP was very high in normal cognitive normal non-Nigerians, not in plasma. Yes. Do you have any other markers that could be characteristic of normal centenarians? For instance, in plasma or in CSF.
So probably I think
other markers apart from the genetic marker that you have said.
So we do have work from Linda, who she has actually looked at. Oh, maybe. Well, maybe you can tell you can summarize the work, her work more. But we do have a PhD student who actually does look at plasma biomarkers. Yeah. So plasma biomarkers in the centenarians and how that differs, because of course the proteomics, as Anna mentioned, there's clearly a distinct signal, but you kind of need to be dead for that, right? Otherwise we wouldn't see it. But maybe you can summarize a little bit of Linda's work, which is maybe more practical.
Well, so what we actually see is that when you take, for example, MMSE and epigenetics, so there's epigenetic clocks like GrimAge, for example, and NFL, plasma NFL, those are all independent markers for increased longevity. And so they don't really associate with each other, but they independently predict the longevity in these cognitively healthy individuals. So,
which is something you can get from blood. So you could,
and MMSE, of course, and so this is the GrimAge clock based on methylation in peripheral blood cells. So these are all independent aspects that would predict for something like this. But what we also see, and if you guys were there at the ADPD meeting, then that the optimism that is happening in these individuals is really surprisingly present in many of them, and which is uniting centenarians across the world. So that's the reason why I started to collect hormones, because you need a specific tube to collect hormones, because otherwise they will unfold. I've never looked into it, but I think that there's a lot of information in the endocrinology there that allows these individuals to maintain so optimistic throughout their whole life. And I'm not sure if that's also genetic. I wouldn't be surprised. This is something that they've had their entire life. So yeah, to what extent that would also explain a good way to age. Well, we'll have to figure that out still. Yeah, but it's truly there. Yeah.
My last question, very simple. Do you think that the copathology in the centenarians is similar to the copathology that can be found in the 60s, 80s, 90s, or it's different? The spectrum.
Yeah. Well, I mean, definitely for these centenarians being 100 plus and having either little amyloid, tau, T43. I mean, so for example, we in the ROSMAP cohort, right? That's a completely different cohort with different selection criteria. And clearly, what you see in there, the selection criteria was at 65-year-olds, you got to be healthy, not demented free, and then you include in the study, and then they just tracked them down over years. So their cohort, in that specific cohort, was more of a natural aging, right? So how does copathology actually look in a natural aging cohort? And then, of course, we can contrast to our centenarians. We definitely see that Lewy body is significantly less prevalent in our centenarians. We definitely see TD43 is significantly less prevalent in the centenarians. So there is something to say about these centenarians getting to that stage, having very little copathology, which is very much different from what we see in the general population, right? I think probably if you think about an age continuum and you start to look at 80-year-olds and 9-year-olds, right? And then you start to segregate, okay, which 80-year-olds and 90-year-olds, which ones are going demented or staying cognitively healthy, that probably starts to maybe align closer to the centenarians. And for those 80-year-olds and 90-year-olds that are starting to become non-demented, that probably starts to deviate away from the centenarians. So I'm not sure if I've answered that question, but yeah, we clearly see a difference if we just look at different cohorts with different selection criteria. But it's definitely at least on the centenarians, it's definitely on the side of minimal copathology. Not always, but definitely minimal.
Yeah, we see very little of it.
Yeah.
Yeah. There was one question in the back. Yeah. Mm-hmm.
Hi, thank you so much for the insightful lecture. Very interesting findings. Not to take a lot of time, but I was just wondering whether you've looked into demographic characteristics, especially pertaining to the resilient group. I was wondering whether besides the genetic protection, they have some other type of advantage, let's say, in terms of social or demographic characteristics for education or lifestyle or diet, for example. And a small curiosity to add to that, I saw that you don't really collect neuroimaging data for your cohort. Is there a particular reason? Is it because of practical reasons or mobility issues that these people might have, or what's the exact reason? I'm just curious. Thank you.
Well, for the neuroimaging part, yeah, definitely there's a partly is practical. I mean, it's kind of a bit hard to do this for 100-year-olds. We did do this in the beginning. I think we had a few.
We did 7 of them.
Yeah,
but 100 something is they'll be fatigued pretty quickly, and you really have to to go through all of the steps together with them, and it was actually very tedious to do. And also they're not patients, so you have to get them in and list them as a patient, and it was a little bit more difficult than we expected. But this was also in my cup of poison time. So now that there's all this genetics data and that that you've worked on, I think people will become more interested and will be more helpful in getting the centenarians in. And I'm hearing a lot of noise now around me that why should we not do this imaging, and we should do this and that. Yeah, I think it's a good idea.
So
but help me, because it's something that I cannot do by myself. I need to use the clinic and all the clinical people that are there. It's very, very costly also.
Yeah.
So if they would contribute to it, then let's do this again. But I think indeed, as I said, with my cup of poison time, that people were like, oh, should we really do it? And I had to push everything really hard. And now I think I will have to push a little less.
Yeah.
So, yeah.
Yeah, and maybe on that note, I mean, there's one of the reasons I'm here is because, of course, you guys also have a brain bank. I think the selection criteria is more on the demented side, but it's still something interesting to see. For example, do we see similar effects or similar mechanisms on the opposite side of the spectrum, right? On the dementia spectrum. And we don't have a lot of data on neuroimaging, but one easy collaboration that we can do is correlate the genetics with the neuroimaging. I mean, you guys have tons of that data. I mean, I've sat together a bit. I've overheard you guys talking about it. I mean, I think we'd be very curious about that type of association, and maybe more on the demographic sides. I'm not sure whether you ever formally made these distributions, but I think like the summary overall is these centenarians are indeed in general are highly educated. Like just for the time of when they were alive and then the era relative to that population, they're very much on the highly educated side. They're not necessarily the most fit either, though. They're a bit
Well, they're fitter than most of the centenarians,
but they're not like historically running ultra marathons and
yeah.
things like this.
Yeah.
And then of course, personality-wise, the positivity, right? Yeah. Really the positivity in life. At least from the genetic side, of course, even though the Netherlands is a small country, there is still different ancestries. We do correct for all these associations. We always adjust for genetic ancestry. But yeah, on the demographics, the other demographic side, we looked at the social, the education bit of.
So they're highly educated.
Yeah.
So are women. Of course, most of them are women in this cohort, and even the women are highly educated, even though these were all born before the Second World War, which was definitely an issue back then. So even then, they were highly educated. They are from a high socioeconomic status, and they had more children than the individuals from their same birth cohort, probably because they were able to afford it. The men were, most of them were smokers before. This is also the reason why we in the Netherlands have very little men who are 100 years old, because most of them did smoke and died because of it. About 70% of the difference in mortality, we call this the gender mortality gap. That's huge in the Netherlands, basically because males started to smoke when they were about 13, 14 years old, and then one after the other. And if you look at the mortality statistics, they start to really go apart, where the women became much older than the males because they all died because of COPD, all sorts of, lung cancers. It was a huge problem. This is now becoming a little bit, it's a bit more the same because women started to smoke more. So that's actually a little painful to see. And the fact that cigarettes are still, you can still buy them, I find that, I don't know, painful. If this is how society works, then, oh my goodness, but it is. Then they did drink very similar amounts of alcohol compared to the people who were born in their birth cohort. They are not the thinnest individuals that you would expect them to see, because I really thought that they were going to be very, very thin because of the restriction, the
diet
caloric restriction. But that is not how the centenarians reach their phenotype, because they eat their cakes and they drink their drinks, and even when we visit them, lots of cookies and lots of lovely things.
I mean, several of them, they like their tarts, they like their liquor.
And they
Some of them swear by it, by the way. Like,
and
really,
some of them still smoke. Actually, when we come in, it just, So there is something about their physique that allows them to resist all that damage that is induced by these lifestyle factors, that allows them to be at this part of the distribution. And of course, it would be interesting to find out what that is. I mean, if we would understand how we could survive all of this smoking, then that would be something very, also very valuable, right? So that's something that I'm also very interested in. And this is something that we haven't published. I would like to really do this again. But if we look at the brothers and sisters of the longevity of the brothers and sisters of the smokers, and we compare it to the longevity of the brothers and sisters of the non-smokers, then the longevity of the brothers and sisters of the smokers is longer, is higher. And that suggests that if you become a centenarian despite the smoking, you must be built in such a way that you are able to, to really process that damage and become healthy again. And that is something probably genetic, because your brothers and sisters also have that. And therefore, so there must be some genetics associated with it. That's maybe not even too polygenic. So something worth to look
It
into.
definitely seems to run in the family.
Yeah,
And
yeah.
how much, environmental, but also genetic, likely.
So yeah, lots to be found out still. So yeah.
Okay, I think, yeah, we will leave you. Beth, thank you very much. And I think we have a lot to do together in the future. So in the near future. So yeah, this is a good way to start it. Okay. Thank you. Thank you very much.